to the die, baby, you'll realize. Baby, you'll see the funk inside of me. Baby, you'll see that rhythm is the key. Get with it, can't think, quit it, stomp on the street when I hear funk blue. Playing pop pipe, follow her to shoot, baby, just sing about the groove. Sing it. The groove is in the heart. The groove is in the heart. Groove is in the heart. Groove is in the heart.
Come on, y'all are crazy, man.
There we go. Hello, everyone. Thank you so much for coming out to join us and being here at STAT at ASH, our event at ASH, as it would suggest. This event is called Standing on Medicine's Threshold. I'm Damian Garde. I'm a reporter at large here at STAT. I hope you've all gotten some food and drink, which is available to you. We have some exciting interviews lined up. We'll be diving into what's next in the fields of hematology oncology, and beyond, frankly. We would love to have questions from you, the audience, throughout the event this evening. Just raise your hand. Either myself or my colleague, Catherine McPhail, will be wandering around with microphones. Don't wait till the end of a given session if you have a burning question. We want to make it as interactive as possible, and we want to hear from you. So don't be shy. Before we get to it, I did want to say, just on the STAT events universe, we are excited to announce that we recently launched registration for our spring summits, the STAT Breakthrough Summit East in New York City on March 19th, and the STAT Breakthrough Summit West in San Francisco on May 19th. You can find more information on each of those on our website. Before we get started, I wanted to give a quick thanks to this evening's sponsors. Our presenting sponsor is McKesson, and our supporting sponsors are GSK and NMDP, and of course, to thank you all for joining us this evening. Okay, so let's kick it off. To begin this evening's program, on behalf of our supporting sponsor, I would like to welcome Dr. Jeffrey Aletta. He is the Senior Vice President at NMDP and Co-Scientific Director of the Center for International Blood and Marrow Transplant Research. Jeff, where are you? Oh, there you go.
Thank you very much, Damian, and thank you everyone for attending. I didn't know if this is a karaoke hour or like a stand-up comedy, but what I'd like to do, though, is share a very impactful story. And I wanted to start with our organization. The NMDP is a leader, a global leader in cell therapy, and its mission is to be able to provide cell therapy for all patients in need. And it's that phrase, all patients in need, that I want to talk to you about. So I'm a pediatric bone marrow transplant physician. And what that means is, obviously, we take kids who have cancer to a bone marrow transplant. And it all starts with a donor. So we have to be able to identify a donor for all of our patients. But the problem is a mathematical one. We know in our registry that there is a low likelihood of finding a donor for patients who are ethnically diverse. And if our mission is to create an equal opportunity for all patients in need, that's our problem to solve. And so the way that we've looked at the problem to solve is by using an old player in terms of oncology, a drug called cyclophosphamide. And we're using it in the context of being able to prevent something called graft versus host disease, and we'll talk about that in a second. So what we know historically is when we used mismatch unrelated donors, we can actually find those donors for patients. But historically, the type of graft versus host disease prophylaxis that we used resulted in adverse outcomes for patients. So literally, your identity of your immune system is HLA, human leukocyte antigen. It's like your driver's license. It identifies your immune system. And historically, we wanted to make sure that every label on your driver's license actually matched another person's driver's license in order to identify you as a donor. But in order to do that, we know that our registry is limited in terms of the amount of donors to be able to provide that. And so we were thinking about using mismatch unrelated donors, but we know that for every level of mismatch, there's actually a 10% decrease in survival. So that 8 out of 8, that well-matched donor, going down to 6 out of 8 results in 20% decrease in overall survival with the regular type of graft versus host disease prophylaxis we were using. That's where the post-transplant cyclophosphamide comes into play. So first of all, we had to see if this was safe and feasible in patients. And the way that we did that was through a clinical trial called the 15 MMUD trial. So our bones on the inside of them have our blood-forming stem cells, hematopoietic stem cells. So when you touch your arm, on the inside is or the outside is the cortex, on the inside is a spongy layer called the bone marrow. And the stem cell literally is like a boat at the dock. It's tethered to the dock and stays there. In order for it to leave the dock and enter into the bay and ultimately into the river and ocean, that's the analogy of a stem cell mobilization. It enters into the blood vessel from the bone marrow, and we can collect those cells. So we collect stem cells through the bone marrow itself, through the peripheral blood, and through umbilical cord blood. And so what our clinical trials did was we first started with bone marrow stem cells through 15 MMUD. 80 adult patients who had high-risk hematologic malignancies, leukemia, lymphoma, or myelodysplastic syndrome, received a mismatch unrelated donor bone marrow product. And we found success with the use of post-transplant cyclophosphamide as graft versus host disease prophylaxis. But we also know that in order to go in and give a bone marrow harvest, a donor has to go to the operating room. And that's obviously a procedure for that donor. So the most common hematopoietic stem cell is collected through that mobilization product that I mentioned, peripheral blood. So that's where the ACCESS trial came into play. We used peripheral blood stem cells that we collected from donors who are mismatched to the recipient and found that that too was successful in terms of a 76% overall outcome. Now that doubles the outcome in terms of what we typically saw historically with that other graft versus host disease medication. So what does that mean? What's the impact story? The impact story is our registry right now, if we're using or looking for matched related donor or unrelated donors, you have a wide spectrum of a match likelihood. So for non-Hispanic white patients, that's an 80% likelihood of finding a match. But for a black African American patient, it's only 29%. But we know by going one level of mismatch, that 29% goes to 84%. We know that another level of mismatch to 6 out of 8, that goes to basically 99% chance of finding a donor in our registry. That is impact. That is hope for patients. And that is the difference that we make at NMDP and are fortunate enough to be able to do that. That's our impact story. Our impact story is making a difference for patients. And right now, when we looked at our data in the ACCESS trial, we looked at those patients who received less than 7 out of 8 mismatch. Those are highly mismatched donors. And we found that they had a similar outcome when we used or compared that to donors who received that 7 out of 8. And even comparable to 8 out of 8 when we looked in our database. So going down to 6 out of 8, we can provide a donor for every patient in this room. And when you look around, we're all diverse genetically. The genetic profile of America is only increasing. So if we want stem cell transplant to continue and to be able to be a viable therapy, we needed to hurdle the HLA barrier, and that's exactly what we did in terms of mismatch unrelated donor transplant. So I'm excited to share this news with you, And Dr. Antonio Jimenez from the University of Miami has a or actually an oral presentation on Monday afternoon where he can go into the details. But let me take a step back. Not everyone understands what a hematopoietic stem cell transplant is. So we want to make sure that we look and review that the data that was seen from this trial, we could check the box in terms of relapse and graft versus host disease, and also in terms of non-relapse mortality, in other words, dying from a transplant. There were equal outcomes from patients who are highly mismatched with those that were not highly mismatched. That's impact. And that's the message that I wanted to provide today. So thank you very much for your attention. And again, always go and think about going to Dr. Jimenez's abstract presentation. Thank you.
All right, thank you, Dr. Arletta. So here to join me for our first session of the evening, please welcome Dr. Yusri El-Sayed. He is Senior Vice President and Global Oncology Therapeutic Head at Johnson & Johnson, and Dr. Laura Grunbaum, Chief Scientific Officer and Senior Vice President of Research at Blood Cancer United. Thank you. Thank you both for joining me. I know I said this before, but I'll keep saying it. We would love to have questions from you, so don't be shy as we talk. But I think the framing that we had for this session was that hematology has been medicine's breakthrough foundry, that so many major discoveries and catalytic technologies trace their roots to the field. And Laura, maybe if I could start with you, why? Why do so many roads seem to lead back to the blood?
I think it starts with the basic biology. It starts with the fact that in the case of blood cancer, you have cells of the immune system that become malignant. And that creates just a very unique situation, challenging, but also an opportunity to really look at, to really understand the immune system, how to leverage the immune system, and how to potentially be able to target particular targets in multiple ways, both in terms of targeting the cancer cells themselves, as well as targeting immune cells that can support the eradication of the cancer. I think beyond that, obviously, blood cancer, the accessibility of samples, the ability to understand and study cell types, the access is something that has greatly contributed. I think we've seen a lot of innovation start in blood cancer, thinking about even the earliest work starting in World War II and post-World War II, exploring chemotherapy, combination chemotherapies, transplant, of course. I think the learnings that came from the early transplants, autologous transplants, later allogeneic transplant, all these have greatly informed, of course, our understanding of the immune system and of oncology, and have allowed us a unique way to really gain knowledge and then transfer it to other areas. And of course, we're also seeing things go the other way now. So I think what I see is sort of there's something cyclical happening where there's innovation happening in one field, in one disease area, and then it's going somewhere else. But I think blood cancer is uniquely positioned because we have this ability to study these cells, and because we're really studying both the malignant cell and the healthy immune system, or how the malignant environment affects the healthy immune system at the same time, and we're just learning so much more.
I wondered, Yusri, kind of segueing from that, the multi-directional things going on between hematology and fields outside of it, how does that work within J&J? You're a hematologist and you were running the hematology side of the business, now doing oncology, J&J, big company, big pipeline. Is there like an information superhighway by which hematology is informing things elsewhere in the business and vice versa?
I think we just actually start applying it more. Even in academia, they always understood that hematology is the best place to understand the disease. Sometimes it's the best place where you understand, the system much better. This is where a lot of things has started. Targeted therapy, understanding the immune system and manifestation of the disease. And in our company, for example, a lot of recent successes was in the hematology space, in multiple myeloma, in leukemia, in other diseases. And we learn a lot from that, and we are applying it now to solid tumor. There's always, it's going to be a learning process, it's going to be more challenging to do, but we are finding that what we learn in blood diseases is actually can translate and can actually lead to very good drugs in the future. And some of these has been, for example, I'll give you an example of the CD3, redirected therapy, which is all started in either lymphoma or multiple myeloma. Now they are moving in a very large way in solid tumor, based on the learning we all learn together from multiple myeloma and other diseases.
So kind of piggybacking on that, Laura, I was wondering, is there anything that kind of keeps you up at night as to impediments to that progress? Like the trajectory that you just described of going from what works, for example, in hematological cancers to solid tumors and then maybe beyond into other diseases, what are the roadblocks that might stop the momentum that we've seen so far?
I think to some degree it's that traditional training often will keep you particularly in one disease area, and I've seen that in my past life in the pharma industry, and I was always somebody who was very curious, applying learning from one disease area to another. But sometimes it's a little bit harder because there's a trend to sort of think about separately what you do in immunology, what you do in neuroscience, in neurological diseases, and neurodegenerative diseases, and in autoimmune diseases. But clearly what we see is a lot of immunological learning. I mean, the fact that antibodies actually became such a huge therapeutic modality, which really came out of our understanding of the immune system, and that the ability to leverage antibodies to leverage immunomodulation, which very much was a big part of the anti-TNF era, understanding cytokine function, et cetera, receptors, and that then moved into both viral infections, chronic viral infections in particular, and into oncology. And that meant suddenly people who were very focused on using chemotherapies, using, in the case of oncology, and some of the direct-acting antivirals, now also had to learn immunology. And I think, so we need people who have that flexible training. We need to maintain the basic research that allows to train and keep that pipeline going, to have people who have that basic training and have that open mindset to really look at these applications across many, many different areas. I think it's gotten much better than it has been in the past because we've seen these success stories, but it's something that we need to continue to invest in.
Well, Yusri, your career kind of fits the theme as well in that you began at the National Cancer Institute and then kind of followed the promise. Well, tell us about your experience with Velcade, which is a sort of path-breaking drug in this space decades ago, but how that led you down this path.
Yeah, so I did my fellowship at NIH, which for people who know NIH 20 years ago, we used to take care of patients at the National Navy Medical Center. And as a new fellow, the worst day in your life is your first day in fellowship, when you are on call and a patient comes into your emergency room, and that's exactly what happened to me. A nice gentleman flown all the way from Hawaii because he has a diagnosis of multiple myeloma, and I have to deal with him. He was one of the most fantastic people I have ever come across, very nice gentleman. Within 2 months, he was dead. Myeloma spread, had renal failure, and died. So that stayed with me. And during my next year, there was this drug called Velcade, and people were reporting that there was a complete response in multiple myeloma. That doesn't, that is fantastic. So after I finished my fellowship, I tracked where that drug went, ended up with J&J. So I have been there for 20 years. I worked on Velcade for many, many years. And since that time, we have now 6 drugs in multiple myeloma in the market. We feel so proud to say that with all these drugs that are available for patients with multiple myeloma, patients should not die from multiple myeloma. They should die from something else. We have drugs now. We have a combination with Darzalex, one of the drugs that we developed for patients who are transplant eligible. Their estimated progression-free survival is 19 years. So that 20 years ago was 2 years. So within 20 years, we have moved from 2 years to now people should die from something else. I think that is the kind of journey we are hoping to translate from multiple myeloma into other diseases as well, and in solid tumor where the unmet medical need is much higher right now.
Well, and Laura, what do you think the pathway is from what he just described and all the transformational things that happen in myeloma to where they could be extended to solid tumors and conceivably beyond into, as you mentioned before, autoimmune disease?
Yeah, that's an interesting question. I think what often has happened that an initial success makes us a little bit overly optimistic. I think we've certainly seen that with the checkpoint inhibitors, right, with the early approvals, and it's been much more challenging to follow up on that and understand better, which patients respond and also how to develop additional agents to further improve these therapies. And I think that is in many blood cancers and solid tumors, of course, still a challenge that we have patients who have extremely good responses, but we don't necessarily have good therapies or even do completely understand why other patients, depending on the cancer, could be a minority or a majority, do not respond. So I think what that is telling us, first and foremost, is that we need to continue to understand, to work on our understanding of the immune system. I think there are some things that are clearly very different in solid tumors. The complexity of the tumor microenvironment, the fact that we often have a necrotic core, that we really need to understand the spatial architecture, and that we really need to understand tumors at the single cell level. I think these are all, and this is not a complete list, but these are some of the challenges that have come up. And I think to me it is always really important to consider that anytime you develop a new therapy and you have patients who are on therapy, do not respond, do relapse, you're looking at different biology now. You may be looking at completely novel biology that you haven't seen before because you're changing not just the cancer cell, you're changing the tumor microenvironment. And so if we want to stay ahead of the game, we have to very early on focus on understanding how this interplay between cancer cells and cancer signaling and the immune system is being changed. And I personally think that the combination of targeted therapies and immunotherapies is probably what has the best likelihood of giving either truly long-term remissions, and if we're lucky, maybe it's going to give us cures. Yeah.
I was going to ask actually, you see that exact thing. Within multiple myeloma, the treatments that J&J has advanced have people using phrases like functional cure as being something on the horizon. How do you look at that? What is the time frame by which you might be able to responsibly say we are curing patients of this disease?
I think we try to be conservative about it because multiple myeloma, like for many decades, was defined as incurable disease, and we said, and there's a new guidelines that are going to come. Hopefully, was supposed to be published today. I don't know if it came out or not. Defining what is functional cure is number of years without disease and not on treatment. I think there is a very large percentage of patients who currently probably are in functional cure with the drug we have right now. If you look at the drugs that have moved the needle in multiple myeloma, from antibodies like Darzalex, for example, or some of the bispecific or CAR-Ts, all these drugs have very significant improvement in progression-free survival, and also some of them have survival data, but they are still, given like very in an isolated way or independently. The bigger cure is going to come on when we figure out how these things are combined together or sequenced in a way, going to what was just mentioned about like different mechanism of action. You put them together, you have a much bigger problem. For example, this was recently released as a press release. It's going to be a late breaker on Tuesday. You put a Darzalex, which is an antibody works through like an ADCC, CDC, and you combine it with a CD3, you are bringing a lot of immune modulation in a regimen. And the data is, you saw it in the press release, with positive survival, positive PFS with a hazard ratio of like 0.17 or something like that. So I think this is where we are going. How do you create regimen? And you take from like 10 or 20 or 25% functional cure now to 70 to 80% in the near future.
So Laura, I wanted to get to the work you do at Blood Cancer United, which includes getting behind and investing in things at, I was going to say the bleeding edge, which is a horrible pun. I won't insult anyone with it at a blood cancer or at a hematology conference. But what are you looking for when you're kind of being the advanced scout of the potential next big things in hematology?
And we're doing investing in a more general sense and also in the literal sense in terms of actual making equity investments and partnering with innovative biotech companies. And what we're really looking for is really related to what I said earlier, to think ahead and to really look at what are going to be the unmet needs, not based on the therapies we have today, but the therapies we're going to have in the near future, because that's what you need to do to really be ahead of the game. we have the huge benefit, of course, that we fund a wide range of academic research, including very basic research, that gives us a very good understanding of the potential new approaches and some of the growing, knowledge about disease mechanisms that is still in very early stages, that allows us sometimes to have a unique perspective when we see a new biotech company or we can support potentially a spin out. So we're really looking at companies that have the potential to be first in class and to address key unmet needs. So these are, typically patients who still relapse after multiple lines of therapies and from even experimental drugs that are in late stages of development, but also very small patient populations, orphan indications. Blood cancer, sadly, is full of those. We're making an investment specifically in pediatric blood cancer, because unfortunately the many approvals that we've seen since around 2017, especially for targeted therapies for adult blood cancer, we haven't seen that in pediatric cancer, and so that is definitely a strong need. So it's really novelty, the ability to address key unmet needs, and then we're also looking at the ability to impact multiple indications, because again, anything that is really truly a groundbreaking new mechanism should have that potential to address within blood cancer, beyond blood cancer, many, many unmet medical needs.
So, Yusri, I wondered, kind of getting to the hematology being a foundry within J&J, which is working in Alzheimer's disease and in immunological diseases as well. How much kind of like cross-pollination is there? Is there like a tumor board style thing for like heads of R&D? Like, how much do you feel like the advances in various aspects of research are informing things within the pipeline?
Well, the hematologists always lead, so we kept leading and bringing them into new places. For now, you see like there's a big shift towards some of the drug and some of the mechanism of actions that work in hematology. I mentioned CAR-Ts or bispecifics. The data in autoimmune disease is fantastic. The same drugs that we used actually for multiple myeloma, if you treat a lupus patient and you deplete the abnormal B cells or reset the immune system. So that's where you see the field is moving now in autoimmune diseases a lot in the same direction. CAR-Ts work in MS, they work in a lot of other neuroscience. So I think different other therapeutic areas are learning a lot, not just oncology, learning a lot from what happened in hematology, and they are applying it.
So, Laura, I wondered, what's the biggest advance you've seen in the research maybe in the last year or so, or the most exciting kind of twist in terms of breakthroughs in hematology that really sticks out in your mind, if there's one?
Well, I have to say it's in the area of immunotherapy. I think the bispecific antibodies, the engagers, starting with the T-cell engagers, but it's certainly not limited to T-cells, is something that clearly shapes across many blood cancer indications and provides also something which is, again, near and dear to our hearts at Blood Cancer United, a therapy that's accessible to more patients, that has really the potential, the more we learn about it, to go out into community settings. Again, ultimately, that's something we need, and we know that that's been more challenging with CAR-T. We're seeing a lot of progress as well. We're understanding side effects better. We also understand how to develop next generation agents that do not suffer from the same toxicities that we've seen, in the early days. But I have to say, I think the Engager platform, and then, we're starting to get a hint of in vivo CAR. It's, I'm cautious here because we've had a lot of optimism about this for a long time, and it has been kind of a long time coming, but I am encouraged that we're now starting to see some data, and this is again in the multiple myeloma space. Interestingly, even though people started out doing it really more in the lymphoma space originally, where we're starting to see some credible data, and so I'm really excited to see where that is going.
I wondered, Yusri, on the subject of CAR-T, J&J obviously has been a leader there as well. The data, or the promise, I should say, the clinical data that has been established with CAR-T makes clear that for many of these cancers it is unparalleled therapy, but it has, less so maybe than a decade ago, but still kind of been hampered by access. It is a difficult treatment for many patients to get. What do you think the long-term horizon is for cell therapies more broadly, given those access issues, and tackling being able to get people who maybe don't live near an academic medical center, for example, to be able to harness them?
Yeah, I think that why our commitment was really to, make multiple different good options available to patients. And some of it is what is really happening now in the market. And this is something I struggle with in my job because I always get asked, why do you need like 6 drugs for multiple myeloma? Why can't we just live with one? Because of exactly that. CAR-Ts is fantastic, is curing patients, but it's not going to be available everywhere and for everyone. And I just mentioned the data that is going to be published or presented on Tuesday as a late breaker, which is a combination of a bispecific and DARZALEX. The data efficacy is exactly like the CAR-T efficacy, but this is an off-the-shelf. It's going to be available for patients to use it whenever they want. So now patient will have an option, and if you can access the CAR-Ts and available and geographically, insurance-wise, access-wise, there could be differences. But if we focus on providing multiple options for patients so they can make that decision with their physician, I think that is the best we could do for them.
Laura, I was going to ask you, you mentioned before one of the potential barriers to continue these advances is people being siloed and there not being investment in the kind of basic research. I wondered if you were presented with someone who was looking to go down an MD, PhD track, or even in business development and industry, how would you make the case for hematology? That that's something they should really pay attention to?
Well, I think it's really one of the best training grounds I can think of, because again, you're learning the immune system, you're learning, a lot of oncology. Of course, the benign conditions also incredibly informative. We've seen, this is an area where gene therapy has already made a huge impact. So it is really a field that provides you a very thorough and very, very broad training ground, no matter where you want to go later. I think that's what we need to do. Of course, we need to ensure that we continue to invest in funding the careers of these scientists. This is something that is very near and dear, again, to our hearts, and that we focus on. And that, young, emerging scientists are really interested in continuing their career, both in academia and in industry. Of course, almost all industry scientists start out in academia, and I think it's really important that we keep that pipeline going.
Well, maybe you've been in exactly this position, but how would you advise the 2 decades ago version of yourself if they hadn't had the formative experience that you had?
Well, I think it's just, most of physicians who go into oncology in the U.S. at least is a combined program, so they get trained in both oncology and hematology. There are very few places where is one or the other. Actually, NIH used to be one of them. And they accept you either at NCI for oncology or NHLBI for hematology, and they can't let you do both. And we actually revolted against that, and we said we want to be trained in both, because the oncology piece of it is where the unmet medical need, but everybody understood that there's a lot of deep science and a lot of things that you learn in hematology that will allow you to go into academic, research. So I think that is still now in our company. We have a very large postdoctoral group, for example, internship and co-op. People come to learn the basic of hematology and how that manifests in different type of diseases. So we encourage that, and I think people who go through that process, they end up really doing very well, regardless of where they end up within hematology or general oncology, hematology, or other diseases as well. So we emphasize that because for one reason, of course, also we have a very large hematology franchise at J&J, so that helps. But I think it's a good practice to learn that basic in hematology, and you can expand from there.
Is there a question from the audience? Yeah, please. Someone will find you with a mic. Wait 1 second. We have a microphone here. And if you wouldn't mind introducing yourself too.
Yeah, I'm Jean-Julien. I'm from Canada. I'm a critical care doctor. Radiopharmaceuticals kind of revolutionized the certain solid oncology spaces, and you would think that being so fragile, hemato-oncology cells would be sensitive to it. Why haven't they taken off more in the heme space? And the second question would be allo CAR-Ts. Why is there a future there?
Maybe I can start. So I think it's all really is the unmet need, and how are you addressing it? So in our portfolio, we have radioligand therapy, we produce a lot of actinium, we have a lot of programs. But if you come and say, in a multiple myeloma with a TCE, I can get you to this complete remission and enhance your immune system, it becomes like very difficult to argue for a radioisotope treatment, which is more complex, and there's aspect of it. In places where the TCEs or CAR-Ts are not working yet, we are developing some of the radioligand, but I see it going to become more and more difficult to justify it in some of the heme spaces. In leukemia, in AML, for example, there's a place for it still, but depending on what else is available and what else can give you that.
Laura, in 10 seconds, do you have an opinion on allogeneic CAR-T?
Yes, I think, the jury's still out. I think for now, again, in vivo CAR, we don't know yet exactly where that is going. I think one space where I think allogeneic certainly could be preferable potentially is when it comes to T-cell malignancies, where I think in vivo CAR will be much more challenging. I think there are also going to be, certainly in the nearer term, real opportunities, because as we know, accessibility, whether it's manufacturing failure, patients' T cells are just not, healthy enough to really lead themselves to good CAR-T production. Patients who can't wait through that production process, et cetera. I think there definitely is a space for allo. Exactly where it is, what it is something that I think we're going to have to figure out. But we're seeing this with companies really trying to get a look at how to position themselves with things like the Alpha-3 trial, for example, for consolidation. So again, there's a benefit to not having to go through the manufacturing process, and that is a significant burden on the patient. So I do think that there is going to be space, but exactly where it is something we'll have to figure out.
Well, that is all the time we have. Thank you both for joining me. This is really great. So next to join us for a discussion on behalf of our presenting sponsor, McKesson, please welcome to the stage Jason Hammonds, President of Oncology and Multi-Specialty at McKesson, and Nathan Shumway from the Texas Oncology, a practice in the U.S. Oncology Network. I don't know what the etiquette is, but I'm already sitting down. Good to see you. So, this fits in really well with a lot of the conversation that we had, because the community oncology is the means by which so many people actually interface with care and with these innovations that are so important. And Jason, I know that's where you guys are doing a lot of work. Can you help us kind of orient us on what does that work look like?
Yeah, so maybe to take a step back from a McKesson perspective and talk about how we support community oncology, because it's probably one of our largest priorities today. Our oncology multi-specialty segment is really focused on two things. It is focused on helping to improve access to care for patients so they can, no matter where they live, they can get access to the highest quality of care, to your point, around community oncology. And the second is helping to play a role in accelerating the development and the delivery of the next generation of therapies. And to really enable that, we've developed a platform of assets that supports every stage of the cancer care journey, all the way from early drug development to the last mile of care delivery. And that's really built on partnerships, and partnerships with community providers, and built partnerships with biopharma, and particularly with our community owned providers. U.S. Oncology Network has been foundational in helping to do that, because it is really, for us, given us a deep partnership with community physicians to see on the front lines with them, what are the opportunities, what are the challenges, and then how can we bring our assets, resources, and capabilities to really unlock that for them, to help them not only be able to provide great care, but be able to expand and reach even more patients close to home.
Got it. So, Dr. Shumway, I wondered, what does that look like on the ground in Texas with Texas Oncology?
Well, at Texas Oncology, our vision is to be the first choice for cancer care. What that means to us is we want to deliver personalized, evidence-based care to our patients close to where they live. And we've been using strategic partnerships and technology to help us achieve that vision. And just to set the picture and paint a picture of Texas, we have close to 600 physicians in our group, and close to 300 advanced practice providers. We have over 300 sites of service. Last year, we saw over 70,000 new cancer patients. 95% of cancer patients in the state of Texas live within 70 miles of one of our clinics. So we're really passionate about getting cutting-edge research and therapeutics to patients where they live because we're such a big, diverse state.
Got it. Well, and you kind of touched on this before, the barriers that exist to this care is geography, I mean, all of these things that kind of come to play. What are we, I don't know, how are you addressing those? What else remains kind of to be done to fulfill that mission you're describing?
Well, I think, for one, community oncologists, I think our role from our perspective is helping to empower them to have what they need. So I think one of the things that we've tried to do is invest in community oncology providers so they have everything to be able to provide state-of-the-art care. I think probably the biggest thing we see today, or I would say, the exciting thing about oncology, it's a little bit of a paradox now in the sense of we're seeing more advancements in therapies than ever before. But what comes with that also is a lot of complexity now that practices and providers have to manage. And so I think one of the things that we've, our way in focusing and supporting community oncologists has kind of always been evolving because the challenges have evolved. And I think right now one of the big challenges is just keeping up with scientific innovations, the amount of burden that's on them. And so we have tried to look in ways at how we can help solve that. And a couple examples of recent things we've done. I mean, one thing that's really important to our practices and us is clinical research. So around 3 years ago, we created our partnership with Sarah Cannon Research Institute to help amplify and elevate the clinical research program that we've had. So that allowed our practices to have deep menus of trials for their patients in the community. But it didn't stop there. As we got further into it, it further highlighted, the challenges that we know in the day-to-day, identifying patients, the administrative burden associated with that. So we've invested technology through things like Genospace, which is a clinical trial matching tool to help drive that. So those are examples, and they obviously cascade through standard of care and other elements that we work to do. But those kind of give some examples of how we are trying to help solve that issue, which I think, keeping up with the complexity is one of the big items right now.
I'll just add to that. I think, when I trained in the early 2000s, we approved less than 10 drugs a year for oncology. This year in 2025, we're on pace to approve close to 50 new drugs. So that explosion of new drugs is wonderful, but trying to get that information out to the providers and to our patients, especially when we're spread out and busy seeing patients all day, is difficult. And so to address those barriers, we've partnered with Ontada, with McKesson. In our EMR, we have AI-powered tools like Care Value Plus, which helps us determine the right treatment for the right patient at the right time. We also have open evidence now integrated into our EMR, iKnowMed, and we're able to use that on a daily basis to help our providers get real-time answers to some of their questions. On the patient side, we've partnered with a company called VidScript, which allows our physicians and nurses to create short videos to send to our patients about their new therapy, about their visit they're going to have with us. It's really helped decrease anxiety and give them education at the level that they need it. And finally, in such a big geographical area, we don't have pharmacy support on site at every one of our clinics. And so we have a tremendous asset with our pharmacy solution teams with McKesson that work with our virtual pharmacists. So we have virtual pharmacists throughout the state that work remotely that can give that care and attention to our patients in remote areas. So all of our oral oncolytics, for instance, we'll make sure that the patients get their drug. We'll check in with them so they know the toxicities and make sure that they're doing well. And we have very robust pharmacy support. And you really can't take care of cancer patients in the community setting without good pharmacy support. So that we've really leaned into that for that barrier. Okay.
I wondered also the clinical trials aspect. When you talk to sponsors, pharmaceutical companies in oncology, they will often say the biggest frustration they have is that such a small percentage of cancer patients participate in clinical trials. It is so difficult to enroll. You've heard all this before. I wondered how what you're describing kind of fits into that problem.
I mean, that is one of the reasons why we're so passionate and committed to this. I mean, I think the reality is that has been the norm. And a lot of that's because I think over time that a lot of patients rather may have to travel to academic medical centers or drive to travel to these trials, we want to bring those trials into the community. So we've got a lot of work to do, but I think that's one of the elements on that. And the other reality too is, kind of goes hand in hand a little bit with Dr. Shumway was saying on the amount of new therapies that are coming to market. These trials are also becoming smaller. Many more trials, smaller patient population. So that's making it more challenging to find the patients at the right time within the window. So, that's why we think the technology, that's why we think the central support we provide. And that's why we also think getting this out to our entire network is so important to be able to fill those gaps.
We know that clinical trials sometimes don't reflect real-world patients taken care of in the community. So we're passionate about providing clinical research in the community. We have close to 200 clinical trials open for our patients in Texas Oncology. We have 54 sites for research within Texas Oncology. And with our partnership with Sarah Cannon and McKesson, we're able to bring that technology to the patients where they live. I'm not sure what the solution is to increase enrollment. It's tough. One of the things that we're working on is how can we get complex therapies in the community setting. We've partnered with a company called Canopy that does our ePros, patient-reported outcomes. They're also able to do remote therapeutic monitoring. And one of the things I've been tasked with doing is trying to get bispecifics throughout Texas Oncology in a meaningful way in the community. We know that these therapies are complex, they've got unique toxicities, and to have the ability to have real-time data and communication between the patient and physician using technology and AI is going to be a game changer. So that's one way that we're leaning into that. The other thing that we have for clinical research, we have something called just-in-time trials. So if you have an orphan diagnosis, a rare diagnosis, or you have a patient that has an odd or a unique mutation, and we have a clinical trial for that, we don't have to maintain that clinical trial apparatus at every site of service. We can open it at the site of service where the patient is within 5 to 7 days with these just-in-time trials. So it's really allowed us to take these patients who otherwise might have to drive several hours to an academic medical center, give them a clinical trial close to home. And so we're chipping away at it, but it's been a big issue for us.
Well, with the few seconds we have, I wondered if you had to leave the audience with one thing that gives you hope about the future of equitable cancer care, what would that be?
Well, I would say, over half of, kind of a little bit where you started, over half of patients today in the country are treated in the community oncology. And what gives me hope is that innovation is not confined to academic medical centers or other areas. This is now out in the community. And we're seeing it through our partnership with practices like Texas Oncology and through our U.S. Oncology Network and all of our oncology ecosystem. And so that helps to make sure that no matter if someone lives in a rural community or whatever, they're able to get access to high quality care. And that's really equity in action. And that's what gives me hope. And I think we're making a ton of progress, but there's so much more to do.
Awesome. Thanks, guys, so much. Appreciate it.
Thank you.
All right, next I would like to welcome to the stage Dr. Bethany Samuelson-Banow. She is the incoming editor-in-chief of ASH Clinical News, director of classical hematology at Cleveland Clinic, and co-chair of ASH's Women in Hematology Committee. And to moderate the discussion, my stat colleague, Catherine McPhail.
Dr. Banow, thanks for joining us today. For those of you who don't know, Dr. Banow is the new editor-in-chief of ASH Clinical News, and you are the first classically trained hematologist and the first woman to lead the publication. As a researcher and clinician, you also focus on patients with bleeding disorders and also focus on treating women and pregnant people, and we're so glad that you could join us today. So thank you for being here.
Thank you. I'm thrilled to be here.
So to start us off, I wanted to ask about this very cool moment that you had a couple of years ago. You actually had a study that went viral on social media. You were testing the absorbency of menstrual products and found that the results of your test were inconsistent with how the products were being advertised, which could lead to the underdiagnosis of heavy menstrual bleeding. But what really stood out to people was that this study was the first known study to test the absorbency of menstrual products with real blood rather than a thinner liquid like water or saline. So first off, I'm hoping you could just take me back to that moment when you discovered that your study had garnered all of this attention and really what it was like to see everybody's reaction.
Yeah, it was a crazy and unexpected moment. I actually got a text from my partner in this kind of nonprofit space that we're trying to create to improve awareness around women's health and periods, and she goes, "You've gone viral. You've gotten all this attention, and all these people are talking about it online." And it was a really telling moment for me because that was not something I expected to be like a crowning achievement, and it was almost a little bit of an afterthought because what we had intended to do and what we ultimately did was look into the use of how many times you have to change like a menstrual cup or some of these newer products to determine if you have heavy periods or not, because all of the old metrics are based on pads and tampons. And then we said, well, but we don't know how much the pads and tampons hold. So I guess we'll do this study before we do the other study. And honestly, I was just thrilled that people were talking about periods. Like that's the win. Like the more we talk about it, the more knowledge there is and the less likely we are to miss these diagnoses. So I took it as a win.
Absolutely. People were mad. They were very unhappy to hear that it had been saline this whole time.
Yes.
I guess tracing that study to today, I'm curious if that moment has had any influence on your work since then.
Yes, it absolutely has. The number of opportunities that has opened up to me has just been mind-blowing. And the interest of the general public has been so encouraging to me in my work because I'm a hematologist. I'm not a gynecologist. And in the hematology space, people don't think a lot about periods. They don't talk a lot about periods. And you try to get a funding for a study about periods, and people kind of scratch their heads. But I have this proof now that people really value it, and there is a space for it. And so it's been just an encouragement to me along the way.
A lot of your research has been looking at data for female-specific outcomes, which there is a real lack of in hematology. When it comes to those blind spots in the data, how is that showing up in your work as a clinician, and why is it so important to address that?
Yeah, so I think a great example of that is one of the direct oral anticoagulants, so these have been around since about 2011, 2012, has higher rates of heavy menstrual bleeding than the others. And that wasn't something that came to anybody's attention until they had been on the market for like 8 to 10 years. Because it wasn't, and the data would have been there all along. We could have collected it. But you have all these women, and it's coming through in these retrospective studies and kind of other ways, and then you kind of have to backtrack and try to recreate it. And to me, that's just such a poignant example of this is a bleeding complication of a medication that causes bleeding, but because it comes out of the vagina, we decided not to look at it. So it is what it is, but we're making positive changes.
I think also the elephant in the room here really is the major cuts that we're seeing to NIH funding and the blacklisting of certain words and rhetoric around diversity and equity. So what impact does that have on your work, and how are you sort of processing and responding to that?
Yeah. I mean, honestly, it has a huge impact on all our work. I don't think there's any part of science that is untouched by this. When we talk about science and we talk about good studies, the first thing we talk about is this data generalizable? And so if you're only doing research on subsets of the population because there are barriers that we are aware of or otherwise to including a more diverse population, we all lose. That's the bottom line. And I hate that we are spending time and effort trying to figure out how to wordsmith things instead of focusing on the science. It's a waste of brain power. And it's really unfortunate. And I'm really proud to work with an organization like ASH that has been so openly advocating for rectifying some of these wrongs.
What is it specifically that you're seeing from ASH that you feel like we really need right now?
So ASH has done a number of things. So I actually gave a little talk to trainees earlier about kind of they're focusing on people, they're focusing on resources, and they're focusing on advocacy. And so this Fight for Hematology campaign is huge and overarching, and a lot of it involves funding, right? Bridge funding for researchers and folks who might have lost their funding or not had opportunities for funding. And that is huge and that is key, and I think that's gonna buoy us up for a while. But I also really love the effort that ASH has put into advocacy and putting things out on social media and making statements, submitting statements to the government about the negative impact of some of these policies, and has really pulled in, hematologists as a community to get a little bit loud about this. And I think this is a time in history when we should be getting loud.
I think another thing that clinicians and patients are really struggling with more and more has been the spread of misinformation and medical mistrust. Patients might come to you with the wrong information, or there might just be a lack of data to answer their questions, or maybe, if you're working with an immunocompromised person, they might be worried about the world around them. So I'm curious, for you in this very challenging moment, how are you navigating that in your own practice?
Yeah, it's really tough, and I don't think there's one right answer. I always take a deep breath and kind of remind myself, where some of this is coming from and what the person in front of me is going through, and really just try to, the only way to fight misinformation is with accurate information. And so I think we have to arm ourselves as much as possible with that. And I think that there's an art to the delivery of it as well. If you are dismissive of their thoughts, they're going to shut down really quickly. And so I think a good principle in life is to get curious. I'm curious why you think that. I'm curious where you heard that. Well, let me tell you what this study shows, and does that change what you think? I think we have to be really thoughtful about our approach.
I'm curious to hear your thoughts. We are living in this very, very complicated moment for science. On the one hand, there are attacks on funding, attacks on science, but on the other hand, we're also seeing some really incredible breakthroughs, some really amazing new medicines in hematology. How are you sort of thinking about the world? Are you worried? Are you hopeful? Where are you at right now?
Yeah, that is a great question. And I find myself often thinking of, this might surprise you, the serenity prayer, right? Grant me the serenity to accept the things I cannot change, the courage to change the things I can, and the wisdom to know the difference. There are a lot of things I can't change right now. I have a toddler, so any of you with a toddler know how that goes. But there are a lot of things that I can change, right? I can interact with the patient in front of me. I can work with ASH on Fight for Hematology. I can talk about periods in rooms like this and with my Uber drivers and all of these other places. And so I'm just focusing on the things that I can change and focusing on the hope that comes from that. And, we're not going to go down without a fight. I have a lot of hope for organizations like ASH and scientists and the community that we will continue to do the good work and spread the word about it.
Thank you. Do we have any questions in the audience from anyone? I also wanted to ask you, the classical hematology workforce has been understaffed for a while, long before the federal funding cuts. What do you think needs to be done and communicated to get people into the field?
Yeah. So my current stance is that classical hematology has an image problem, and we got to work on it. Maybe we need like a PR rep or something like that. But people get the impression like, oh, you can't make a career out of that. Let me tell you, I will never be unemployed. There will never be a day when there are not more patients than doctors in classical hematology. So I think we need to let people know about that, and I think we also need to get them exposed to this earlier on, right? If it's an afterthought after you've spent 6 or 7 months on the oncology wards, then it might be a little harder to envision yourself as a classical hematologist. I think spreading the word about these exciting, like we're having gene therapy, we're having, potential cures for some of these classical hematologic diseases, and it's a really exciting time, right? You can cure a disease that somebody's been born with and would otherwise die with, and you can't say that in a lot of fields. And obviously, I'm like really passionate and a total geek about it, so be that as it may, but I think a lot of us really, really love it and really enjoy it, and I think we can share that enthusiasm. It'll help.
What would you say to someone who is worried about the state of entering science as a whole? Like, what is it that is really keeping you going these days?
Yeah, I think the work is more important now than ever, and you can't underestimate the enormous sense of purpose that I think comes with science. And everything worth doing is hard, right? Like I don't think there's none of us has an easy life, right? That's just the reality of it. And so you kind of got to pick your heart, and you got to do it scared. And I have never regretted my choice, and I don't think I ever will. And I think most of the people that I know in science and in classical hematology are very glad to be here.
When we spoke backstage about things in hematology that are inspiring to you, you specifically mentioned your heroes really keep you motivated. Could you tell us more about who those people are and the lessons that you have learned from them?
Yeah, I have a lot of heroes. Dr. Avalos, who is our current president of ASH, comes to mind. I had the privilege of getting to know her and hang out with her at a week-long training institute that we did for young researchers in hematology. And the work that she has done and the leadership that she has shown in the fight for hematology has been so inspiring to me. And just, she doesn't get tired, she doesn't give up, she just keeps going. And I think that is very inspiring. I have a lot of peer mentors who I think are just absolutely incredible people, and getting to watch them and celebrate their successes and see the differences that they're making is hugely beneficial to me. Our trainees and students are heroes to me as well, because they're making this choice in a time when, the future isn't clear, and they're still going after their passion. And also, I would say my patients, very brave souls, very kind and thoughtful and wonderful people who deserve the best care that we can offer. And at the end of the day, I think that's what keeps us all coming back and doing more.
I also wanted to ask you, we were just talking before about some of the new medicines that are coming into the field, and I guess I'm curious from your perspective, both as a clinician and a researcher, when you're looking at something like a gene therapy, which is, there's a real discrepancy between the innovation and the access of these drugs and the data that's available to your patients. How is that playing out for you in this moment? What do you hope the future will look like?
Yeah, well, I hope the future just keeps getting better. When I think about, when I was entering into fellowship, right, the DOACs were coming out, and that was the biggest and kind of the only thing really in classical hematology that had been new in a while. And since that time, we've had such a revolution, right? We've had longer acting factor products, and we have, treatments for TTP that act quickly, and we have, the rebalancing hemostasis and the gene therapies. And so, we're getting there. I think, with gene therapy in particular, for me particularly in hemophilia, I think it's going to get a lot better in the future, so I'm actually not too sad that we have a little bit of time to stew on it. But I hope eventually we'll get there. I think the realities of the payer system that we have kind of disincentivize, I think, coverage of some of those products. And so, we'll see if and how that changes over time. But things do tend to become more affordable over time, and so that's the hope I hang on to.
Thank you so much. Just want to check again, any questions from the audience? No? Okay. My last question for you is, if you're talking right now to somebody about the field of hematology and science, and they're thinking about entering it, what is most worrying? What's exciting? What do you think the future is going to look like?
I mean, I think to me, what's most exciting is it's fun every single day. It's such a great job. We get to make diagnoses. We get to work with patients long term. We get to offer cures sometime. There's a huge amount of variety that I really love, and it's really exciting. And I think we're starting to hear more talk about classical hematology. ASH has certainly made an effort towards having, more of a focus on classical hematology. So I think all that's very exciting. I think, the worrisome thing, there's always something to worry about, and they come and they go and they cycle, and, nothing lasts forever. And so I just like to keep the focus on, like, we're in a period in time. It's not eternity, and if we just kind of put our heads down and do the work and are focused and encouraging, then I think we'll get through it together. And I can't imagine a better field to go into, and I would certainly encourage any young person who had interest to pursue it.
Thank you so much, Dr. Bano. I really appreciate your time tonight.
Thank you for having me.
I'll let them go. All right, thank you, Catherine and Dr. Bano. Up next from our supporting sponsor, please welcome to the stage Nina Moyash, Senior Vice President for Global Product Strategy and Oncology at GSK.
Thank you very much. I definitely have wrong shoes for standing and walking on the stage, but I'll do my best. So I will talk about curiosity. That was mentioned a few times before me. Curiosity is something that I think everybody involved in drug development is very familiar with. Translating science into clinical and medical practice requires curiosity continuously. But I will talk about slightly different aspect or point of view on curiosity, one that requires a bit of humility and that tries to answer the question, what did we do wrong when we failed? My name is Nina Moyash. I'm the head of oncology global product strategy at GSK. And some of you might be familiar with a drug that GSK is marketing in multiple myeloma called Blenrep. Blenrep is one of the very few, very rare medicines that has an interesting path. It was approved and then withdrawn and then approved again. It's a path that is unusual and also highly uncomfortable. When I was put in a place of taking the accountability of relaunching Blenrep, I didn't really feel the usual excitement one would feel when you're launching a brand. And I asked a few questions. And first one was, Jesus, why me? And the second one was, why bother? Why are we doing this? And I was not the only one asking that question around me. Within my own organization, there were people who were really saying, well, really, Nina, do you want to do that? And why don't you just give up? But those were not the only voices. The other side of the voices that we were hearing were coming actually from the community physicians. From the community physicians and from the patients who were treated in that community setting. And hearing those different voices made me wonder, what is it that we did wrong? And what is it that makes them ask us not to give up and to continue working on the drug? Multiple myeloma is a relentless disease. It's a deadly disease, despite number of options that have been approved. Not all the options are fitting every patient, and many patients continue to die of multiple myeloma. Listening to patients and physicians in the community, what we have heard is there are no other options, and they want a treatment that will fit their life rather than life fitting their treatment. And one of the patients that we talked to said something that stayed with me and actually ended up on many of our materials. She said, we want more life, more years in our life, but we also want more life in those years. And that really describes what patients want. When we launched Blenrep the first time, we were focused on traditional metrics, regulatory endpoints, efficacy, response rates, PFS, hazard ratios, p-values, all the things that we were very much used to. We spoke our language, but we realized that we didn't really speak patients' language. And that curiosity of hearing different feedback, hearing different opinion, led to us really humbly talking to those community physicians and patients who are treated in that setting. And what do they need? They want an option where administration, drug medicines can be administered in their community practice. Physicians want an option where they can do it close to patients' home. And they want an option, as I said, that fits the life rather than life fitting the therapy. It's quite humbling and really a lesson that requires a bit of courage, driven by huge level of uncomfort. But just a few weeks back, we got a message from one of the patients, Kathy. She's been on Blenrep now for 5 years. And it's, I think many of us have heard these stories of patients who have been on therapies for a long time. But her message was not only about 5 years, it was about her life in those 5 years and everything that she managed to do and how it was, I don't want to say easy, but how her life didn't really change. And hearing physicians and hearing patients giving those stories is really critical when you need to be brave and even relaunch a drug that nobody really believes in. Thank you.
Thank you, Nina. So for our final session of the night, please welcome to the stage Linnell Hoke, President of Cell Therapy Organization at Bristol Myers Squibb, Cindy Peretti, Executive Vice President at Kite Pharma Inc., and to moderate, Adam Feuerstein, Stat Senior Writer for Biotech.
Hi, guys. I want a big shout out to the Stat event team for transforming a sports bar into a hematology oncology meeting space. You should have seen all the work they had to do to get the mechanical bull out from the middle of the thing there. That was like a lot of work, so hats off to those guys. Linnell, hello.
Hello there.
How are you?
Great.
Cindy?
Doing well.
So it's kind of cool. Between Kite, Gilead, and Bristol, we have probably the three largest, most successful cell therapy companies. The little secret here is they're also competitors, these two companies here. So we won't have them at each other's throats quite yet. We'll get to that point. But I wanted to just start kind of with the theme of the evening, big picture. Obviously, you guys are both significant players in the autologous CAR T markets, which really has been transformational for patients and obviously also commercially successful. And I just, kind of big picture, where are we right now in CAR T for cancer, and how much bigger can we get, do you think, with the existing products? We'll talk about some of the innovation and some of the next generation type stuff, but with the products you have now. Linnell, why don't you start?
Yeah, first of all, a great tee up. I think Cindy and I would both agree that autologous CAR T has been transformational. It has set an efficacy bar that very few modalities have even come close. And when we think about over 50,000 patients have been treated with autologous CAR Ts across multiple different targets, multiple different diseases. And so really where we are today is how do we continue to advance the field on a couple dimensions? First and foremost, the autologous CAR Ts that we have available today, only 2 out of 10 patients who are eligible to receive them are getting them. So the first order is to how do we ensure access to these transformational modalities? Number 2 is how do we continue to look at additional targets? Because there are some patients, despite having transformational modalities in earlier lines, are progressing. So looking at targets like GPRC5D, so we're quite excited about that target and bringing a CAR into that space. And then the third dimension is how do you bring CAR T into other disease areas which you couldn't imagine bringing a functional cure to? And I think about autoimmune. I mean, it's hard to imagine just 3 years ago, a conversation with someone with severe lupus would never, ever include that I'm going to potentially put you on a trial where we may be able to give you a deep, durable remission. You'll have no active disease and be off all your immune modulators. I mean, that would never be thought possible. So when I think about, CAR T, where we are, in some ways I'd say we're kind of at one point of the innovation curve. In other parts, I think we're just at the beginning of CAR T. And then I think I forgot to mention just to say, and of course you can't help but say, I think the full unlock of cell therapy, and I know Cindy also has got a lot of exciting things in their pipeline here, is off-the-shelf solutions. So looking at HDALLO, which was mentioned earlier, as well as in vivo CAR T. So we're all excited about the potential of bringing an off-the-shelf solution that can actually deliver on the same efficacy that we see from autologous CAR T.
Cindy?
I think that was nicely said. I think the pieces that I would add to that is that getting more than 2 out of 10 is really important for us. We know today in multiple myeloma, because those referral patterns exist into the academic centers for transplant, we're seeing 4 out of 10 patients. So it's possible. And on the lymphoma front, we're doing a lot of work, all of the companies, to really move the needle to make sure that more patients get this. This is a one and done, potentially curative treatment, and we need to get it out there. As Linnell suggested, there's a number of things coming down the pipeline, and we're convinced that you also need dual targeting. And so our ability to look at hitting 2 targets and really improving the efficacy level for patients and maintaining or even having a better safety profile is going to be critical. We're excited to share some of that data here at ASH. And we think the same for multiple myeloma, solid tumors, autoimmune. It's going to be important to hit multiple antigens.
Sticking with lymphoma, you mentioned, there's just a lot. It seems like there's a lot of patients out there that are not getting CAR T therapy right now, lymphoma patients. And we've heard about the barriers before. We've heard manufacturing as a barrier, academic versus community center setting as a barrier. What is this going to take to sort of get? So instead of 2 out of 10, we're at 4 out of 10, 5 out of 10. What do we need to do? What do you guys need to do to get there?
I think we both would agree it's not a manufacturing issue anymore. That was when we all initially launched. We had difficulty being able to take a very personalized medicine one-to-one and scale it. But that issue has been overcome. And now we have unlimited supply for our patients across the manufacturers to be able to provide these patients. And so really now it's about looking at what are those barriers on getting this modality to the community. You heard the panel talk about it earlier today. Patients want to be treated closer to home. And so our ability to bring CAR T into the community where they are being maybe initially diagnosed or at least being treated closer to home is really important. And I know both companies and the industry has a big push to say CAR T, now with the REMS and the removal of some of the burdensome patient label, as well as the burden on the treatment centers, is affording community practice to challenge themselves to say, hey, listen, I'd like to bring CAR T in the community. And we're excited to say that we've got many centers now that are true community practices, not academic centers with an outpatient center, but true community practices that are delivering autologous CAR Ts.
Let me ask you about that. If you're a community center, you're interested in bringing CAR T to patients there, what are the challenges that the community center faces?
I think that there's a few things. Community centers often, because they're not large hospitals, don't have apheresis units. And so that's something both companies have worked to overcome. How do we provide apheresis for these community practices so they don't have to build up those expertise and they don't have to be a hospital to deliver it? Then you think about the infrastructure to deliver a therapy like CAR T. Maybe you need a -80 freezer. Maybe there's a few things you need. I think those are lower hurdles. Really getting each piece of what it takes for that patient's journey solidified with those community practices is what we're all working on. And when they do it, and they are doing it today, they recognize that it's not as hard as they thought it would. They're having conversations with their friends, and it's pieces like that, I think, that allow us to really get to the community. The last piece is you talked about the REMS. It's removing hurdles like that. There is FACT accreditation often required for private payer reimbursement, and FACT has been working with a number of industry partners and with the community in creating a modular certification that you can do in the community that doesn't have the same level of need that's in the accreditation in an academic center. It's every one of those pieces that's going to help us unlock it.
I want to ask you about I think for a while there was always this debate between autologous and allogeneic. Obviously, autologous, patient-specific was out there, and there was this conventional wisdom that allogeneic was going to eventually come in off the shelf, boom, 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 easy. That has not happened. Many companies focused on allogeneic have shifted to autoimmune, and we'll talk about autoimmune in a second. But there's a couple companies that are still out there doing it. Do you think that is still something that is viable, or have we now just sort of like leapfrogged to in vivo, and that's kind of the next frontier?
I think it was said earlier tonight, I think the jury is still out. What I think the jury is not out on, the first generation HG allo assets really struggled with persistence and potency of the cells. It was clear. And so what needed to advance was the gene editing techniques to be able to have these cells not to have the same exhaustion they would have and have the persistence, particularly in hematologic or oncology indications. And so what we're excited about at BMS is we actually are doing collaborations with next generation gene editing, like companies like Prime Medicine, and so we actually are growing in our confidence that HG allo can work. We have an asset that will be coming early next year into solid tumor, and you have to be able to deal with the tumor microenvironment, and you have to have the persistent potency of those cells. So we're excited to try these things. I think in vivo CAR T is another extraordinarily exciting field. It was mentioned earlier today. We are obviously equally excited, and we too are going to let data play out. And for me, I think having multiple modalities, multiple targets, gives you the opportunity to meet the needs of multiple patients. And that was also highlighted earlier today. And I think BMS is uniquely positioned as one of the only companies that's got shots on goal across autologous, HG allo, as well as in vivo CAR T across hematologic solid tumor, as well as autoimmune. So we're really excited about
we'll
the
get
future.
back. I want to talk in vivo in a second more about that. But Cindy, let me ask you, so if I had, if we had Caribou or Allogene up on stage, they would say that they've solved the durability issue. It's just a matter of execution, their clinical trials, but they feel like they are competitive. They're both allogeneic, off-the-shelf CAR T for CD19 lymphomas. They think they're competitive. Do you agree with them? Disagree with them? What's your view?
I am more skeptical on allogeneic approaches in oncology for some of the reasons you highlighted, which is the tumor microenvironment. And I actually believe we're going to need multiple antigens. We're going to need armoring to be able to tackle solid tumors. But for hematology, I'm skeptical on the allo approaches. And where I do think allo, I think the jury's still out, we heard earlier, is in autoimmune. And the ability for allo to really get as deep into the tissue as you see with autologous and in vivo right now, that's something that I think we're going to have to wait and see.
So going back to in vivo, Cindy, tell us a little bit about, because you've made some, Gilead Kite has made some investments in the in vivo world. Tell us about what you're doing there.
Yeah, so we acquired a company called Interius that's in Philadelphia, Pennsylvania. We're really excited about it. The construct that Interius has put together, the vector that we put together is a modular vector, so it allows us to very easily move in and out binders and a number of things that we think are going to be critical for in vivo to be efficacious in a way that balances efficacy and safety. The thing about in vivo is the first time we're seeing autologous-like responses in that Lancet paper with the BCMA. And so we're very excited about it. We've also done a strategic collaboration deal with a company in China called Pregene, which allows us both with Interius and Kite and Pregene to rapidly iterate on these constructs and be able to test them in patients. Today, we both know animal models don't work as it relates to cell therapy, and our ability to get into patients and really understand that becomes important. And so having those pieces, in particular the Pregene collaboration, where we can look at patient data fairly quickly, tweak our modular in vivo vector if we need to. We're excited about that, and we think that in vivo is going to bring an off-the-shelf option, lower cost of goods. What we're seeing in the early safety data out there is that it looks really promising. It's safe and efficacious.
And Linnell, you guys bought Orbital Therapeutics. Now, is that for you? Is that an autoimmune acquisition, or is that cancer, or is that both?
It's both. I mean, the lead asset, OTX-201, is an autoimmune asset, and so we're quite excited. We think Orbital brings a lot of differentiation. It's a circular RNA approach which we think uniquely positions you to be able to have a lot wider cargo load, which means it actually can potentially play into hematologic malignancies as well as solid tumor, which was in part why we went after an LNP strategy, because we wanted to be able to move into the autoimmune disorders. You're talking about patient populations that are tenfold hematology. You are going to have an off-the-shelf solution. Almost 80% of patients in autoimmune disorders are treated in the community, not too different than oncology. And so we wanted to get to an off-the-shelf. And so this opportunity with Orbital really presented a really exciting opportunity, because what we saw in the PK/PD data for Orbital matches what we see on CD19XT, which is our asset. So we were seeing what we wanted to be able to see to match an ex vivo CAR T. And if that indeed plays out in humans and ultimately plays out in the field, you're talking about one of the biggest disruptors in all of autoimmune. So we're really excited about moving very quickly there. But I think what ultimately sealed the deal on why Orbital was when we got to look at their construct and its ability and flexibility to potentially move across into heme and solid tumor. It became a natural marriage.
I mean, we're here obviously to ASH, and there's that Chinese company, not Chinese, they're U.S. company, Colonia, right? They're U.S. company.
Yes, they are.
Has I think 3 patients worth of data,
BCMA.
in vivo BCMA
a
CAR
viral
T
approach,
that's being presented at this meeting, and what we saw in the abstract at least was some pretty exciting. So it shows you, I guess you both what's good. It shows you the potential that there is for in vivo.
It does, and I think the piece that we both neglected to mention is you don't have to lymphodeplete. So not only is this off the shelf, but the patient isn't going to go for lymphodepletion, which is fantastic from a toxicity standpoint and allows you to treat the patient earlier. We also are both talking about in vivo, but we have integrating in vivo, and then we have non-integrating LNPs, and both areas are really exciting. We're working in both as well, and I'm excited to see what's possible for patients.
So sticking with the meeting, Cindy, anito-cel, which is the BCMA-targeted CAR T that you are partnered with Arcellx. You have some of the data from the pivotal study. We cannot talk about the number. The data are being presented tomorrow, and because we have to be good little boys and girls on stage, we cannot actually give you the data. It's all
they all
still.
Can't keep secrets.
It's all. There's probably some ASH people out there. If we mention numbers, they'll probably yank our badges. But maybe just give us sort of like the roundabout way of telling us about what we might see tomorrow in the data presentation.
Yeah, we're excited to share the data. So you're going to see more mature data. It's at 15.9 months worth of data. And I think the piece that we're really looking forward to with anito-cel is bringing it to patients sooner. It's really a differentiated construct, and it's not differentiated in any single way. I want to talk about it holistically. The data that you're going to see for efficacy is very strong, and we're showing durability. The data for safety is we continue to see no delayed neurotoxicities, so no Guillain-Barré, no Parkinsonism, no Bell's palsy. We have not observed any enterocolitis, and our rates of ICANS are in the low single digits. And so we're excited to see the safety profile married with the efficacy profile. Then you couple that with our world-class manufacturing and our ability to get this therapy to patients in the same times that we're showing with our commercial products today. With a global footprint, with 570 authorized treatment centers, we're excited to bring this to patients.
And the competitor there is Carvykti, the J&J legend BCMA CAR T. And I'm looking forward to all of the investors and analysts tomorrow arguing about the data and whether you're like superior or they're superior, because that's been an ongoing debate at every ASH or every EHA even where we have these data updates.
It definitely is a debate. I think, again, I'm going to stress the holistic nature of it, of looking at the strong durability and efficacy, looking at the safety, and looking at our world-class manufacturing.
So, Lynelle, it's always nice when you come into an ASH where you get some nice FDA news.
Yes.
Right? I mean, perfectly timed.
Yeah, I designed that way.
Breyanzi had another approval that happened just
today.
Oh, it was like
This
Wednesday,
morning.
right?
This morning it was announced via press release.
So tell us a little bit about that.
No, we're really, really proud of, Breyanzi. Breyanzi launched third to market, and it's now the number one CD19 CAR T in the world. And it was a long journey, and we now have the broadest set of indications, this being its fifth and the only one in MCL. So we're really proud of the work we've done with Breyanzi. Its overall profile, both from an efficacy and safety, is one that we're really excited about. Also, at ASH, we're really excited about our 4-year Transform data. We'll be presenting unprecedented OS rates for Breyanzi in Transform and also in Transcend and follicular with 86% there, and when our Transform data is 61.5%. So we're really, really excited. I mean, you think about why we all got in the business of bringing CAR T, you want to say 4 and 3 years later, you've got a significant percentage of patients that are still alive and surviving. That's why we do the work we do. So we're very, very excited about Breyanzi. We're also excited about Arlocel. Arlocel is an asset that targets GPRC5D. And for those of you familiar with that target, one of the cons of that target is when you put ongoing pressure on that target, there are some unwanted toxicities that are pretty difficult for patients to deal with. That's what's only available today is a TC, which is that ongoing pressure. What the GPRC5D CAR T provides is a one-time hit of that target, and what we're seeing is transformational efficacy for patients, particularly post-BCMA, that also don't have some of those on-target, off-tumor toxicities that are very unwanted by patients. So with all the data that's coming out at ASH, where BCMA is going to continue to climb into earlier lines and expand in its use, I do think this is going to be a unique opportunity with our
Breyanzi came out of Juno, right? Is that where did Breyanzi come out of?
Yes, Juno.
Juno.
Mm-hmm.
So sometimes these acquisitions that pharma makes work out.
Indeed.
Right?
Indeed.
So again, this is where we get in a little bit of the competitive. Obviously, Breyanzi competes with Yescarta and Tecartus, but Cindy, why don't you give us, why should community centers and academic centers, why should they be using the Kite products over Linnell's products?
So I'm going to start by saying I think it's great that there's options for all patients.
That's a very good initial answer. A very safe answer.
Safe answer.
But what I will say is we have with Yescarta and Tecartus, we have very strong efficacy, similar to what Linnell just shared. We continue to share long-term follow-up in both constructs. And we feel like patients who have highly aggressive disease, whether it's primary refractory, high tumor burden, really benefit from therapy like Yescarta, not just from the therapy itself, but also from our reliability and rapid turnaround time on our manufacturing.
And you also have some, you mentioned, I think you did mention the dual targeting stuff, and I know there's some early data, right, that you're presenting. You can tell us a little bit about the CD19, CD20 product you guys have.
So we have a bicistronic construct, and we'll be presenting phase 1 data tomorrow. So it's a CD19, CD20. What bicistronic means is that it creates actually 2 CARs, and both of those CARs are on the T cell. And so we have the ability to get around antigen escape around CD19 with the CD20. We also know CD19 and CD20 are very prevalent in lymphomas. The second piece we did was 2 co-stimulatory domains, CD28 and 41BB. And this is important. We looked at every derivation of this, of having CD28 on both, CD28 on one, and 41BB on the other, and 41BB on both. And what we saw by the way we've positioned it, which is an axi cell with the CD19 and then the CD20 with 41BB, is it allows for the rapid tumor killing, but it allows for a slower, more gentle persistence in what we see. And so that's coming through. Hopefully you'll be able to attend that tomorrow as well, both in the efficacy and the safety that we're observing.
I didn't know if there's any questions out there, Damien? Anyone want to?
Does anyone have a question?
Chime in, anybody. Maybe ask some more competitive questions.
Can I add one more thing on it? Should I do?
Go ahead.
Okay. For the data we're showing tomorrow, we also, for Kite 753, have done the bicistronic with 3-day manufacturing. So 3-day manufacturing means what we're harvesting is more juvenile cells. And so we're able to dose at 1/10 the dose of axi-cell today, and with the more juvenile cells, and see really good efficacy and safety. So
what else do you have at ASH? What's the highlight, the Bristol highlights at ASH?
So the big highlights are Breyanzi and Arlocel. And so, to answer your question before and why we are excited about Breyanzi, I think Breyanzi was made for the community. And we hear that from physicians. It's also why it's the fastest growing part of our business is into the community. So our fastest growth is coming out of outpatient and in the community. And what we hear from community physicians is they want a drug that absolutely is deep and durable and provides them the efficacy they want, which they're excited about that they get from Breyanzi across a broad set of indications. But they also want a safety profile that's predictable, that they can give to a broad range of patients. So I do feel like that is a strength of Breyanzi's. And so you'll see data here that demonstrates that in a real-world setting. So encourage people to go out. There's 2,400 patients worth of real-world data here across France and the United States that will demonstrate that profile. So we're quite excited about that. And in addition, I think the bigger place where we join forces is we also have a publication here putting data behind this 2 in 10 stat. Everybody throws this 2 in 10 stat on, but nobody's actually published on it. We actually have published data that will show from Komodo data, which actually show you longitudinally that indeed these patients not only are eligible for CAR-T and are not making it, but why. And so I do think it's important that we don't just talk about this 2 in 10 stat. We got to put the data out there that it is actually true, and number 2, why it's happening. Because the best way to tackle the 2 in 10 number is to know why and actually amplify the issue in every avenue you can.
I'm going to get out of cancer for a second. Cindy, tell me about Aspen Neuroscience, because it's you guys, Gilead Kite just invested in them. I think you're on the board, and it's a CAR-T, auto CAR-T for neuroscience.
It's iSPC cells for Parkinson's disease, and they have some very interesting data. So we put a strategic investment into Aspen, and I'll be sitting on the board. I think the piece that we really appreciated about it is Parkinson's is a difficult disease, and if you can find therapies like cell therapy that can be a one-and-done treatment for patients, it's really meaningful in these diseases. Autoimmune will be the same.
Yeah, and last time you and I spoke was around the ACR meeting, the rheumatology meeting, where you guys presented some of your autoimmune. I mean, that seems like a big strategic focus for Bristol when it comes to cell therapy.
Yeah, that ACR was a mic drop moment for CAR-T, if I'm being honest. So we ourselves presented over 71 patients worth of data. And to say it was standing room in every session we were in, and the clamoring for rheumatologists to understand more about CAR-T would be an understatement.
This is in lupus, right?
So we had it across several diseases. So we had lupus, scleroderma, and myositis. And the most moving moment was we had our myositis data there. And I've been to a lot of conferences in my over 25-year career, and I've sat and looked and been at poster sessions and seen doctors present poster. I've never seen one of the lines on the graphic walk up to that poster. It was one of our myositis patients. The physician who was the lead PI, it was not his patient. He did not know or expect that this patient would walk up. But she said, I wanted to see when this data was published and when it was in poster, and I wanted people, when they came to the poster, not just to see a line, but to see me. She's a journalist. She was so debilitated. She could not be a journalist anymore. She could not function. When I tell you, when she walked into this room, this beautiful young woman and vibrant again, I said to myself, I'm like, these are the watershed moments where you say to yourself, as many hard days as we've had in CAR-T, gosh, it is worth it. You just get one of those moments, and for us, we're super excited about this next frontier for CAR-T.
All right, on that very hopeful note, I think we will, Damien, I thought I heard Damien. We'll end it there. 15 seconds left to spare, so
wow,
we're good.
Excellent. Thank you, my friend.
Thank you.
Cindy, Linnell, thanks so much.
Thanks.
Thank you.
And thanks to the audience, too.
Thank you, Adam. Thank you both. Okay. I have brief closing remarks. Thank you all for being here. Be sure to check out our website, as I mentioned before, for our East and West Coast summits coming in 2026. And if you are going to JP Morgan this January, we hope you'll join us. We are hosting our usual co-working space exclusively for Stat Plus subscribers. And we will have our annual evening event on Monday, January 12th, which will include Alnylam CEO Yvonne Greenstreet, and we'll have Adam back on stage chatting with a few investors, including Rod Wong of RTW. You can go to our website to register for that. Until then, thank you again to our sponsors this evening, our presenting sponsor, McKesson, and our supporting sponsors, GSK and NMDP, and of course to all of you for joining us tonight. We will have drinks and food for the next 34 minutes, so we hope that you stick around and enjoy yourself. And of course, again, thanks for coming.